Hey guys, it's going to be a very quick video about eukaryotic DNA organization and storage. So our chromatin, if we look deep within our chromosomes, and there's a very good illustration of this on your doc today. If you go down, I think it's about page 2 of the doc where it says chromatin and condensed chromosome structure, you're going to see that our DNA, which I'll show in orange here, our DNA is actually coiled or wrapped around these proteins called histone proteins. So it's a concept that's sort of like this, right? So the orange line there is the DNA and the blue circles are the histones. Now, I've only drawn 3 histones there, but just a side note, and you can sort of see this in the drawing, or at least some of the drawings, 8 histone proteins, right? Let's just say there's 8 there with DNA coiled around it, right? I'm just going to kind of, this is our chromatin, DNA and proteins, boom. One of those units is called a nucleosome. Not a crucial word, but if you do see that word nucleosome, it's just our DNA wrapped around a cluster of histone proteins, and that would make up a little bit of a chromosome. So back to this more simple drawing, what we're going to learn, actually this is a really simple concept. If the histones are close together, the gene can't be read. It's too tightly coiled around these things. RNA polymerase cannot do transcription here. When the histones move farther apart from each other and the DNA gets sort of spread out more like this, and by the way, you're missing out. We do paper doll models of this in class. Oh, so exciting. I'm so sad that we're going to miss it. Let me just wipe away this single tear off my cheek. Okay, moving on. Now RNA polymerase is able to, here's a big blob. That'll represent RNA polymerase. RNA polymerase can now jump on this. It's probably going to find a promoter region in front of the gene that it can attach to, and it's going to be able to zip along and transcribe this gene. So this would be the arrangement of the gene when it's on, and this is the arrangement of the gene when it's off. And that's really the concept here. Now, one detail I'll add is that small chemical tags called methyl groups will bind directly to the DNA, right? So this little pink box here is a methyl group, small functional group, small molecule. Methyl groups will bind to the DNA. So when we see that DNA is methylated, that really means the histones are tight together and the gene is off, inaccessible to RNA polymerase. Another group of small molecules which attach actually to the histones themselves are called acetyl groups. And when acetyl groups are attached, it tends to push the histones apart, almost like repelling magnets, and the gene is now open. So we have here acetylated DNA and methylated DNA. What else do I want to say about this before we stop this video? What we're going to see is that individuals, and this is actually, I think, one of the most interesting insights of the whole entire course, honestly. Individuals, not only do they have their own DNA sequence that is individual to them, unless they have an identical twin, of course, which in that case, that twin would have the same DNA sequence. But for non-identical twins, for people who are not twins, you have your own unique DNA, your set of DNA, your genome. But you also have what's called the epigenome, right? Epigenome means on top of the genome. And that is your distinct pattern of which genes are off and which genes are on. And we're learning that your behavior, your environmental influences, your diet, these types of things can actually turn certain genes on and off in your genome. And then here's a key insight. That pattern, your epigenetic pattern, that was created during your life can actually be inherited. All right, and I want you to think back for a moment to Lamarck, that evolutionary biologist who had that wrong idea. He was famous for being wrong, if you remember. Lamarck was the guy who said certain animals develop certain features because of use and disuse. The giraffe has a long neck because it used its neck more. The crab has a big claw because it used that left or right claw more. That is wrong, and Lamarck is in all the textbooks for being an idiot, basically, when in fact he was a pretty smart guy. We're sort of seeing a little bit of a Lamarckian way of thinking here, that something you acquire during your lifetime, let's say you smoke cigarettes, and that methylates certain genes in your DNA and those genes are off. Your children and even your grandchildren in some cases can inherit that genome that way with those genes off. So we do see in this part of the unit that your behavior, your activity in your lifespan can actually influence the inheritance of your children and your grandchildren, which is sort of scary if you think about it. Okay, I'll stop this video here, but you're going to do a lot of work around this on the doc, and I'm going to do a screencastify for the doc as well.